at work because I had some experiments to do this morning. I had to go and see my cell babies, look after them. So we are back in work and it's Friday, so I've got to make sure my cell babies are tucked up for the weekend and they're happy and I can leave them. So let me know if you are in the chat and you're ready to learn about epigenetics and inheritance today. So this is aimed at kind of A-level, so probably ages like 15, 16 plus, because we are going to be doing some hardcore genetics today. We'll do a little bit of a recap from last lesson as well, because we need to remember what an allele is before we can move on. So, hi Jack! Let me know if you guys are in the chat and you're having a nice Friday. And you'll go back to school soon, don't you? I think, in September. I don't know. So all the uni students come back soon, so Oxford is going to be really busy soon. It's been really quiet at the moment. So say hi in the chat. I just watched one of Nish's videos and I realised Nish has a nickname. He calls himself Nishful Thinking. So I think I need to come up with some kind of biology nickname for myself. Let me know if you've got any ideas. Nice ideas, please. Nice nicknames. So say hi in the chat, let me know you're here and you're watching, because today we are doing epigenetics and inheritance. Okay, so a bit more genes. So while we are waiting for everybody to arrive, because Leonardo was so late last lesson, he missed the entire lesson, okay? So Jack's put, I'm home educated, so it'll probably be a bit over my head, but try to keep up. Jack, don't worry. I'm going to hold your hand and walk you through it. It's easy. Biology is easy and fun, okay? So while we're waiting, there's a hair, while we're waiting for everybody to arrive, I forgot to do a fun fact to start the lesson. So instead, I'm going to show you a picture of my children that I went to see this morning. These are my children. So at the moment, I thought I could show you what they look like. So this is a picture of my cells under the microscope. So these are the cell lines I'm growing at the moment in like a big Petri dish. And this is what they look like. So they have really boring names. So this one is called DU145. Don't ask me why. And this one is called LN-CAP. Okay, that's their name. And they are prostate cancer lines. Prostate cancer. So this is what my cells look like. I went to look at them this morning. They're looking happy. Gave them some food. They'll be fine over the weekend. And then on Monday, I'll come and see them again and make sure they're not too crowded, because I don't want them to look like this picture. There's way too many cells in that picture. I want them to kind of look like in between these two. So like, I want a 50-50 between these two. Because if they get too overcrowded, they become unhappy, okay? So you don't ever want your cancer cells to become too crowded, guys. Top tip for if you ever do cell culture. Cool, so I'll wait a few more minutes before people arrive, because I realise it's only 10:05. Cool. So we need to start off with a reminder. Can anyone remember from last lesson what is an allele or an allele, however you want to say it? And the picture is a clue. What is an allele? Let me know in the chat. Tell me. What is an allele? Correct. Points for Jack and Karina. So it is a variant of a gene, a different flavour of a gene, different variety of a gene, okay? So we all have the gene for eye colour. We all have eyes. And we all have the gene for hair colour. We all have hair. But they just come in different varieties. And remember, you get one from your mum and one from your dad, like this. So one from your mum and one from your dad. And then together they form your pair of chromosomes. Okay? And now this would be my chromosome. Chromo. Right? So you get one allele from your mum. Remember, I think people are quite used to saying you get one gene from your mum and one gene from your dad. But for this lesson, we need to get into the habit of saying you get one allele from your mum and one allele from your dad. Okay? Remember, allele and gene kind of mean the same thing, but it's just a different variety of a gene. So this lesson, we're going to use them interchangeably. Remember, allele just means a gene, a different version of a gene. Okay? Now, on this previous slide, I drew them like two halves of a chromosome. And that's totally fine to do, but in biology, we wouldn't draw like two halves of a chromosome. What we do is we assign each allele or each gene a letter. We give them a letter, okay? So let's say we're doing blue eyes and brown eyes as an example, okay? Actually, I'm gonna do colours to make it easier. So, blue eyes is gonna be lowercase b, and brown eyes is going to be capital B, and you'll see why in a minute. So let's say your mum gave you the allele for brown eyes and your dad gave you the allele for blue eyes, okay? This is how you would write that down. If you were trying to describe what alleles you got from your mum and dad, we use letters, and we use capital letters and lowercase letters to basically assign which allele is dominant or recessive. And we'll talk about that in a minute. So, if you get the same allele from your mum and dad, same allele, so let's say your mum had blue eyes and your dad had blue eyes, yeah, you would have both the alleles for blue eyes. We would say this person is homozygous. This is a new word you might have come across before. Homo meaning the same, okay? So if you get the same allele from both your mum and dad, you are said to be homozygous for that gene. And I'm going to put it in a sentence so it's easier for you to understand. So I have blue eyes. Both my parents have blue eyes. So I'm going to assume Hannah has blue eyes. She got a blue eye allele from her mum, a blue eye allele, can't spell allele, from her dad. She is said to be homozygous for the blue-eyed gene. Okay? That means that I have the same allele variant from both my mum and my dad. I am homozygous, new word. If I didn't, if I got the brown-eyed gene from my mum and the blue-eyed gene from my dad, I've still got a gene for eye colour, yeah? I still have colour in my eyes. If I have a different allele from each parent, I am said to be heterozygous for that allele, okay? I feel this is the best way I can describe this on an online whiteboard. It's quite a difficult concept to grasp, okay? So if you have the same allele variant from your mum and your dad, you are said to be homozygous. If you have different allele variants from your mum and your dad, let's say you've got the brown-eyed allele and the blue-eyed allele, you are said to be heterozygous for that gene. I hope that makes sense. Moving on. Now, I said these two words a minute ago, but I want to see if you guys know what this word means. So what do you think I mean when I say this is a dominant gene? What do I mean, a dominant gene? So that's my first question. Question number one. What do I mean dominant? How can a gene be dominant? Let me know. What do you think? How can a gene be dominant? If you've heard of this phrase before. Jack's put the gene that's switched on or working. Yeah. Alison's put in heterozygous, dominant allele is the one that is expressed. Yes, correct. So the dominant allele is the one that gives you your phenotype. And when I say phenotype, this is your observable characteristics. I'm actually going to do a brand new slide here. So genotype would be this, like brown eyes, blue eyes. So genotype is just your combination of alleles. So genotype is your combo of alleles. Your phenotype is the physical characteristics that you get from your genotype, okay? So let's say the brown-eyed gene is dominant here and the blue-eyed gene is recessive. This person will have brown eyes. This person will have brown eyes because the brown-eyed allele is dominant. So dominant means that is the version of the gene that will always be expressed. I'm going to put that down. So the version of the allele that will always be expressed. Oh, my phone's going off. Sorry, guys. So dominant means the version of the allele that will always be expressed. And you only need one copy. Oh, sorry, put me on silent phone. So you need one copy, all right? So if I go back to this example, I only need one copy of the brown-eyed gene and I will have brown eyes. But why do I not have blue eyes, even though I've got a copy? That's because the blue-eyed gene is recessive. So if we go back to recessive, I need 2 copies of that allele for it to be expressed. 2 copies of the same allele for it to be expressed. And I'm going to use blue eyes as an example. Let me just move this. So, oh, where's my pen gone? There. So, up here we had big B for brown eyes, little b for blue eyes. So this person will have brown eyes because the brown-eyed gene is dominant. But so my phenotype is blue eyes, so I must be little b, little b. I have to have 2 copies of the recessive blue-eyed gene for me to actually have blue eyes. Again, I hope that makes sense. The more you come across these like allele things written down, Jack's put, how do some people end up with heterochromia, 2 different eye colours? That's like an actual genetic condition. I have no idea. I have no idea how people get heterochromia. It's not really my area of expertise. Right, so now we know that a dominant gene is the one that will always be expressed. You only need 1 copy. A recessive allele, you need 2 copies in order for you to have that phenotype. And remember, genotype is the actual combination of your alleles. The phenotype is what you look like in the end. So brown eyes, brown hair, blonde hair, green eyes, okay? That's what your phenotype is. Now, we need to do, like in A level, they always do an example of each condition, okay? Now, in this example here, we are going to use cystic fibrosis. Cystic fibrosis is a really good example of a recessive condition, meaning you need to have 2 copies of the cystic fibrosis faulty gene for you to actually get that condition. okay? Does anybody know what cystic fibrosis is? First question, what is cystic fibrosis? If you've heard of it before. The picture might be a bit of a clue. What is cystic fibrosis? Alison's put it's a build-up of mucus, yeah. So in a healthy person, this is my airway. In a normal person, what happens is there's a mutation in the chloride pumps, I think it is, in the airways, and basically these pumps that pump across ions across your membrane, they don't work properly, so this person gets a build-up of mucus in their lungs and they can't breathe properly, they'll get loads of infections, they need to have physio to kind of like clear away the mucus. So up here, draw your attention here, they've just used F for fibrosis, right? You can use any letter, but for this picture they've used F. So the normal allele is capital F and the faulty allele is lowercase f, right? So parent 1 is mum, parent 2 is dad, okay? The dad has cystic fibrosis. Why? Because he has 2 copies of the faulty recessive gene. Your mum, she has 1 copy of the faulty recessive gene. And what this is this is called a Punnett grid. And this is basically working out the percentage of what children will have the condition. So all we do is work out all the different combinations of what the kids might end up with. So you basically, all you do is you write down the alleles for the mum and the alleles for the dad, and then you just cross them over with each other. So if I was to cross big F and little f, I would end up with this combination and this combination. Oh my gosh, I'm so burpy. But if we were to cross this F and this F, you'd get this combination, and then again this F and this F, this combination. So there's actually a 50% chance, see, of the child being born with cystic fibrosis if one of the parents has the condition. We're going to do more on that later. Now we're going to do the opposite. This is an example of a dominant disease. Remember, dominant means I only need 1 copy of the faulty allele for me to get the condition. Faulty allele, and then I have the disease. Right, what is Huntington's? Does anybody know what Huntington's disease is? Has anyone heard of Huntington's disease? Let me know. You guys knew what cystic fibrosis was. So have you heard of Huntington's? Let me know. Let me get a different colour. So Huntington's disease is a neurodegenerative disease, right? So neuro to do with the brain, and degenerative means it gets worse over time. So in a normal person, we have this, the Huntingtin protein, okay? And this lives in your brain, normal, chilling, healthy brain. But in someone who has Huntington's disease, they actually make loads and loads of repeats in that gene. So normally, we've got less than 35 repeats of CAG. Remember, these are base pairs, genetic base pairs. So normally, we've got about 35 of these that go CAG, 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 right? They repeat themselves in the gene. That's normal. But in people with Huntington's, they have loads of these repeats. So they'll be like, 100 CAG repeats. And what this does to the protein is it makes it really long and tangly, right? It basically causes these tangles, these protein tangles in the brain, which causes your neurons to degenerate. And you get symptoms like tremors and shaking, and you can't control your movements, things like that. And unfortunately, these people die quite young. And it normally presents, I think it presents quite early. So like before you're 40, you start to see the symptoms. So this is an example of a dominant condition. Now, if we look up in the Punnett square, we've got the Huntington's allele as capital H because it's dominant, right? This parent doesn't have the faulty gene. It's a normal person. This person in red has Huntington's. So again, we do the crosses, and again, we've got a 50% chance of this baby being born with Huntington's disease. We're going to do one of these ourselves in a minute, so we understand. Oh, and here we are. Right, so a Punnett grid, and you need to do these for A-level. This is working out, go back, working out all the combinations of alleles possible. We'll put from mum and dad to baby. To baby, right? And you need to be able to do these by hand at A-level. So we're going to do an example together. So in the exam question, let's say the mum will be, and let's do blonde hair, okay? So let's say the mum is BB and the dad is BB. And in the exam question, they will always say little b is blonde and big B is brown. And they will always tell you in the exam question what is dominant and recessive, but you should know with the capital letters. Capital is always dominant, okay? So the brown-eyed gene is dominant and the blonde-haired gene is recessive. So if we want to work out all the possible combinations of what this baby's hair colour is going to be, we need to do a Punnett grid. So what you do is you write out on the top and on the side what the parents' alleles are in a little grid, like a noughts and crosses grid, okay? Like this. Ugh. Laggy pen. Get rid of that, right? And now all we're going to do is do all the different possible combinations, okay? So I'm going to label the squares, 1, 2, 3, 4. So what would I'm going to do square one as an example. So square one, if we cross these two, the baby will be capital B, lowercase b, if that makes sense. So what would number 2 be? What combination of alleles would number 2 be? What combination of alleles would number 2 have? If I'm crossing this and this. Jack's put capital lowercase. And what would 3 be? Remember 3, I'm going to be crossing this and this in blue. What would 3 be? Get it? B. Yeah, Jack's right, BB. And 4 would be BB. Okay? Now in the exam question, they might ask you what is the phenotype of that child, okay? So now I want you to tell me what colour hair the baby will have. So will baby 1 have brown hair or blonde hair? What would the phenotype of baby 1 be? Make sure we use the proper scientific terminology. Yes, brown. Baby 2? Brown. Baby 3? Baby 3, blonde. And baby 4, blonde. Yeah, cool. So this is this is exactly the same as what is on this diagram, yeah? Apart from all they've done here is a disease and all we've done is like hair colour. But if it was a condition, you would say, and it was dominant, this baby would be affected, this baby would be affected, these babies would be fine, okay? Now, one more definition I need to tell you is, what do I mean if somebody is a carrier of a genetic condition? What is a carrier of a genetic condition? Pop it on the board. What is a carrier? Alice Stone, 100 points to you. Yeah. So they have one allele for a recessive condition. So if you're a carrier, this only refers to recessive genes, okay? Let's use cystic fibrosis as an example. Capital F, lowercase f, and this lowercase f is the faulty gene because it's recessive, lowercase. So if I was this phenotype, I would be classed as a carrier because I've got the faulty gene, but I do not show the symptoms of the disease. I do not have the disease. Okay? And if the mum is a carrier and the dad is a carrier, there is a much more likely chance that the baby will be born with a disease because there's more chance of the baby having the combination double lowercase f. Okay? Okay, what's this a picture of? What is this? What are these furry things? What are these? If you came to last lesson, it might have given you a bit of a clue. Alice Stone's put, is it expensive or possible for standard people to have their entire genome sequenced to see everything that's wrong with them? Number one, in the UK, it's not possible. Well, I mean, like, I get my cancer cells genomes sequenced, and it probably costs me about £2,000 per sample. So if I were to get my entire genome sequenced, it would be about £1,000. But I don't know how you would do it in the UK. Like, it's not routinely offered. I know in America, if you've got like a rare genetic condition, they will sequence your entire genome to see like if you've got any other random mutations. So yes, Alice Stone, it is possible. It's about £1,500 for a genome, I think. But yeah, you can do it. I'd like to have mine done. Karina's put, Jack's put teddy bear legs. This is a chromosome. This is the X chromosome and the Y chromosome. Okay, look how enormous the X chromosome is compared to the Y chromosome. This stubby little Y chromosome. Okay, now I want you to remember the difference in size between the X and the Y chromosome. So the X chromosome has about 2,000 genes on it. The stubby little Y chromosome has about 78 genes on it. Okay? Doesn't do much. The X chromosome's got a lot of genes on it. Now, this is really important for this word, X-linked. Because, no, I'm going to describe it here, X-linked. So remember before we said dominant and recessive. Okay, we've got dominant, we've got recessive. So those are modes of inheritance. We need to introduce a third type called X-linked. This means the disease, the faulty gene is not carried on a normal chromosome. The faulty gene is carried on the X chromosome, okay? Sometimes they're called sex-linked disorders, but they're called X-linked, okay? So this is the third type of mode of inheritance we want to do. And this is called X-linked. And one really good example of an X-linked condition is colour blindness, okay? The faulty gene for colour blindness is found on the X chromosome, on the female chromosome, which is really interesting. So again, we're going to do an example. The dad is not colourblind, and the dad is XY because he's a boy. The mum is not colourblind, okay, but she is a carrier. She has the faulty gene here, but she is not colourblind. Now, if we do the whole Punnett grid again and we cross them all together, because girls have 2 X chromosomes, it's basically the probability of them getting 2 copies of the faulty gene is not very common, okay? It's quite rare. Because boys only have one X chromosome, if it's much easier, it's a higher probability of them picking up the X chromosome, the faulty X chromosome, because they only have one X chromosome. It's literally just a game of probability, okay? So because boys only have one X chromosome, the chance of them having an X-linked condition is much higher than a woman, because the woman basically needs to have, you know, the double probability, the double bad luck of picking up the X-linked condition. So X-linked conditions, the rule is they are much more common in men, which is why much more men are colourblind than women. Some people say it's not possible for a woman to be colourblind. It is. It's just much rarer, okay, because she needs 2 copies of the gene because she's got 2 X chromosomes. It's basically easier for the guy to pick up one faulty X chromosome. So, colourblindness is more common in men because the faulty colourblind gene is found on the X chromosome, and because boys only have 1 X chromosome, they're just more likely to get it than girls who have 2 X chromosomes. Cool. And to check you guys aren't colourblind, have a look at this. Let me know if you can see all of the numbers on these circles, okay? And this is called the Ishihara test. It's a test for colourblindness, okay? Colourblindness. Give me a thumbs up or a thumbs down in the chat if you can see all of them. If you can't see, Alice Jones put 25, 6, 56, 45, 29 and 8. Alice Stone, you are not colourblind. Congratulations. Cool. So a person with normal colour vision can see all of the numbers. But if you were colourblind, this is what you would... Jack said, I can only see the first one. Jack. Right, if you are colourblind, this is what you should be able to see. So the most common type of colourblindness is called red-green colourblindness. I'll just put CB, okay? So if you've got red-green colourblindness, this is all you'll be able to see. You won't be able to see any of these ones. Okay? My dad is colourblind, but I have no idea what type of colourblindness he has because he says he can't see traffic light colours. So it's red, orange, green, and my dad thinks it's red, orange, white. Okay? So for my dad, white means go on traffic lights. And he had a bright purple phone for 4 years and thought it was black. So this is the test for colourblindness, Ishihara. What do I mean lethal allele? And why do I have a picture of Peter Dinklage on this slide? If you know the song, Peter Dinklage, Peter Dinklage. Okay, what is a lethal allele, and why have I got Peter Dinklage to illustrate this for me? Let me know in the chat. What is a lethal allele? The clue is kind of in the name. What's a lethal allele? Do 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 do. Peter Dinklage with a crown on. Alison's put, is it an allele that reduces your life expectancy? I can see where you're going with that. So, one example of a lethal allele is, I'm going to spell this totally wrong, I know I am. Let's try. Achondroplasia, which is also known as dwarfism. Okay? Peter Dinklage. If you have one copy of this allele, the embryo will not survive. Okay? So that baby will not be born. That's what I mean lethal. But if you have 2 of the recessive copies, you will be born with dwarfism or achondroplasia. So if you have a copy of a lethal allele, it basically means the embryo won't survive. But if you just have the recessive version of that allele, then you might end up with a condition like achondroplasia. I think I've explained that right. But that's not really a massive bit in A-level. Right. Have I got time for this? Oh, no, I'm going to have to skip that slide. No, I'm going to have to skip that slide. We're going to move on to epigenetics because we've only got 10 minutes left. So what is epigenetics? What is epigenetics? Who's heard of epigenetics? And I'm going to write some words. Right. What is epigenetics? I'm just conscious of the time. Whoa, I've done too much content again. What do you guys think epigenetics means? And epi means above or on top of. On top of genetics, okay? Epigenetics, I'm going to do a definition, are changes in the DNA, but not to the DNA sequence, that are heritable, right? So normally we think of changes in your DNA, so it's just your sequence of DNA that is inherited. But epigenetics are basically modifications of your DNA, but not to the DNA sequence, that can be passed on to generations. So things that we are adding on top of, epi on top of, the DNA. And normally these are extra little proteins, protein groups that we stick onto the DNA somewhere that change our gene expression. One example is histone modification. Remember last lesson we spoke about histones? So those are the proteins that DNA is wound around. Okay, my pen's upside down. This is a histone and the blue is the DNA, it winds around it. We can have what's called acetylation. This is where I add a little acetyl protein or group, a little acetyl group, onto the histone, or specifically the histone tail, okay? They've actually got these little, look at his tails in the air. So histones have these little wavy tails, yeah, that the DNA is wrapped around, and we can add little acetyl groups onto these tails, right? What's the point? What does it do? Basically, long story short, if you have acetyl groups on your DNA, it changes how tight and loosely they are packaged, okay? So, if your histones have acetyl groups on them, they are very open. Your DNA is more loose. Think of it as the loose segments in a slinky, okay? I'm going to put acetyl. So if I have little acetyl groups added to my histone tails, then your DNA will be nice and open like a stretched out slinky. If I have lots of methyl groups, a different type of group, added onto my histone tails, I am going to be squashed together like the squashed parts of the slinky, okay? So, acetyl groups on histone tails make your DNA more stretched and loose. Methyl groups on your histone tails make your DNA like a squished slinky, very compressed. Why is this even important, how loose and squished DNA is? If my DNA is loose, it means it's easier to express a gene. So it's easier to make a gene because I have easy access to the DNA, right? If my DNA is all packed up and squidged tight, I can't get in, right? I can't get in. So I'm going to put can't get in. It's harder to access the DNA if it's all squished together. Hope that makes sense, guys. Time check. Second type of epigenetic modification is called DNA methylation, okay? All I'm doing is adding these little methyl groups to my DNA, okay? Again, what's the big deal? What do these methyl groups mean? Well, the general rule is the more methylation, so I'm going to put more methyl groups on a DNA, it means the gene will be silenced, okay? We won't express that gene. And epigenetics can be influenced by your environment, right? Which I think is really interesting. So, you are a baby in your mum's tummy. Here, this is me. And the food that your mum eats will influence your DNA methylation profile. How insane is that, right? So whatever your mum eats when she's pregnant, this will affect your epigenetic profile, right? So your histone methylation and your DNA methylation, which is insane, right? And going back even one more, your grandma's diet when your grandma was pregnant with your mum will influence your epigenetic profile, right? So epigenetic changes and modifications can be passed on through generations, which is amazing, okay? One really key example is this. This was the Dutch famine, or it's also called the Dutch hunger winter. And it was a time in 1944 when I think the Germans restricted food to, they restricted food to this part of the country. And basically there was a really bad famine. And what scientists found was, they found here that people who were exposed to the famine 6 decades later, in their siblings, they had changes in the methylation profile of this gene, okay? So 60 years after this, children whose great-great-grandparents were in this famine, that influenced their DNA methylation profile. How insane is that, okay? And if you're interested, the IGF gene is involved in making insulin, okay? So, your epigenetic profile is influenced by not only your environment, but your mum's environment, and your mum's mum's environment, and your mum's 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 environment, okay? What's this got to do with disease? So I just put this picture here because I liked it, okay? So you can see the little methyl group added to the DNA, and we can see our little histone tails with their little acetyl groups added, right? But it's not as simple as that, okay? This is a much more scientifically accurate diagram. Now, when we're talking about epigenetics, we have 3 different players on the field, okay? We have writers, readers, and erasers, okay? Now, the clue is in their names. The writers are the ones that add, they write the groups onto the DNA. So they add the groups onto the DNA. The readers, they will come along and read that group. So if I read lots of methylation, I'm going to be like, oh, I won't make this gene. This gene must be silenced, okay? So they read the code that the writers put on. And then we have erasers. They can come on and rub off different marks that the writers have put on. And epigenetics is such a complicated balance between all 3 of these groups. So the writers coming along, and the readers reading, and the erasers rubbing out. And all I want to try and say with this picture is, in diseases, this balance, this very fine balance, is a bit off, right? So we might get writers adding on loads and loads and loads and loads. Or we might get too many erasers, like rubbing off marks where they shouldn't be rubbing off marks. And what I'm trying to say is, we can use drugs to stop these 3 people doing these things, okay? Let me see if I can highlight a good one on the board. Here, okay? So let's say this dude is reading this methyl group on the DNA, And here, these are the drugs we can use to stop him doing that, okay? So in cancer, for example, or lots of diseases, the balance between the writers and the readers and the erasers is a bit off, okay? Which means genes might not be appropriately expressed, and we can use drugs to stop them from doing that. So then maybe we can, instead of this gene being silenced, this gene can now be expressed. I'm done, and I've gone over by 3 minutes. Right. Hopefully that makes sense, okay? Epigenetics is something you definitely did not do in GCSE, and it's something you will come across in A-level, okay? So epi means on top of the DNA code. Epigenetics is adding modifications onto the DNA and histones, and this can be influenced by your diet, and it can be passed on through generations, but we do not change the DNA code. We don't change the base pairs, okay? Hopefully you have learnt very much from Sensei Hannah today. Put it back on the screen. Okay, that's the end of the big catch-up. So I think we're going to be going back to doing like learning adventures. If you missed this lesson, don't worry, you can go back and watch all of them. No worries, Jack. It is very interesting. You are welcome. If you guys have like any questions, put a little comment on the Facebook video or something. But yeah, epigenetics is super cool. I love it. It's really hot and fashionable in cancer research. So yeah, we're going to carry on doing learning adventures. If you've got any requests, please email us. If you want me to do a topic, let me know and I'll try my best. And I hope you have a fun and safe weekend, and you're all looking forward to going back to school because now you know everything about A-level genetics, okay? So have a nice weekend, guys, and I will speak to you very soon. I'll miss you all so much.